honoring the discoveries of 3 scientists that revealed an unanticipated world of small non-coding RNAs that regulate gene function in plants and animals. From their days together as postdocs, Victor Ambros and Gary Ruvkun shared a journey of unanticipated discovery. Following a pathway not seen by others, they were to uncover the secrets that regulate gene activity.
Victor and I worked really closely, just like right arm, left arm, doing this. We were sort of thinking the same thoughts. And so we were doing the genetics of development. We wanted to know how it was that the animal knows to move from a larval stage to the next stage.
C. elegans, the simplest, complicated animal with its 959 cells, presented the perfect research subject for Ruvkun and Ambros to study how genes control development.
We thought that we would just learn more about really interesting principles of developmental timing control. We tried to understand really how this system worked in the worm. We didn't really expect that what we were studying was something really new.
Far beyond their expectations, they discovered some very small RNA molecules which possessed a never-before-imagined impact on the growth cycles of the worm.
The problem was defined by the mutation. Victor and Bob Horvitz realized that the mutations that they were studying where the animal couldn't lay eggs. They realized the reason it wasn't laying eggs is it was never becoming an adult. It kept reiterating larval stages. It was a failure to grow up.
Ruvkun focused on Lin14, while Ambros examined Lin4. They had no idea how sharing their findings would lead to new realizations about the role and relationship of these small RNA molecules.
We knew that the 2 genes were closely related, and the genetic analysis of the genes indicated that they had some kind of intimate relationship.
These genes form essentially a switch, and you can think of developmental events, any developmental events, as throwing switches. So, at the 1-cell stage, when that cell divides, one cell becomes the back end of the animal, one's going to become the front end of the animal, and switches get thrown at every division. The goals of our labs are to figure out how this switch works. How does the Lin4 gene regulate the Lin14 gene?
The moment of miraculous insight came when Ruvkun and Ambros put together their findings.
This was your classic midnight phone call.
And we sort of read off the sequences to each other over the phone. And it's really, really exciting.
Do you see it? Yeah, there it is. And we're sort of pointing out all the hallmarks. And look, these things are pairing. And then we could see that they match up. It was like a lock and key.
So that was very exciting. And it was particularly exciting, as you imagine, because we shared that moment. I think that each one of us would have felt kind of a little bit rotten if the other one had seen it before ourselves.
And the chance of this happening just by chance and that we were getting fooled was something like 1 in a million.
Although Ruvkun and Ambros had entered a new territory with their research, its far-reaching implications were not realized until David Baulcombe, a botanist in England, found similar findings in plants.
What we were trying to do was to genetically modify plants so that they would be resistant against viruses. If we were dealing with a flower pigmentation gene, for example, the gene silencing effect blocked the flower pigmentation gene, and it didn't block any of the other 30,000 genes in the plant, as far as we could tell.
Realizing these RNA molecules could be much smaller than anyone imagined, Baulcombe devised a series of experiments to hunt for them.
And then what we could see was that the silencing effect spread through the plant. So actually there's a signal molecule that associated with silencing that is most likely these little small RNAs that moves through plants. So then we got to thinking about how cells communicate.
From both sides of the Atlantic, the evidence was mounting. The small or microRNA findings of Baulcombe, Ruvkun, and Ambros suggested that small regulatory RNAs existed in many organisms.
We sort of suspected that by then that was some sort of link with Ruvkun and Ambros and their small RNAs.
That finding of Baulcombe's really brought together kind of a package of findings that provided a framework for this hypothesis of Lin4 and Let7 being part of a broader phenomenon.
Early in 2000, after discovering Let7 RNA, Gary Ruvkun did a simple internet search. To the world's amazement, he found the small RNAs weren't just a worm thing, but regulated growth and development in a multitude of organisms.
Not just microbes, not just the worm, not just animals, but plants, and related to RNAi. So it really sets it up as a tiny RNA universe.
So immediately, I at least had to completely reorganize my view of the universe. And so it was just instantly obvious that there had to be more small RNAs.
These regulatory molecules discovered by Ruvkun, Ambros, and Baulcombe are now known to impact a wide range of normal and pathological activities.
I didn't think of it that it was going to be related to cancer, but it is. And it's true of many, many genes, and it's a very finite set. 10,000 genes is not a lot of genes. So there's more than 10,000. That's what makes organisms different from each other. But the core that makes us much the same is a very finite game. And it's essentially playing chess with those 10,000 pieces.
Looking where no one had looked before, Ambros, Baulcombe, and Ruvkun discovered an unforeseen universe of small but powerful RNA regulatory molecules. With this new knowledge, we better understand both normal physiology as well as disease.
By sort of following that trail of curiosity, we ended up discovering a new kind of gene.
And the key to figuring out that these small RNAs existed was that we got a mutation in it. There was a mutation, and the worm led us to the small RNAs. We just followed where it said to go.
It's not due to vision on the part of anybody, it's just we stumbled on something very strange.
We know just the tip of the iceberg about tiny RNAs.